Everyone, thanks for coming. This is a wonderful occasion. We are gathering here to witness Cesar's dissertation defense. Cesar graduated from Immokalee High School in 2016, and he then came here to USF in 2020 after graduating with a bachelor's degree in engineering, or bioengineering from Florida Gulf Coast University, and he's been doing wonderful work here. He'll be summarizing his dissertation in the next 45 minutes or so. Please hold your questions till the end of the presentation. On the other hand, the committee members are welcome to ask if you have any questions to get some clarifications only. Discussion and other stuff we can wait for the later part of the meeting. So the committee members include the major advisor, Professor Chris Pasaglia. I am the committee chair, Shiva Swaminathan. And the other members include Bob Frisina, Ash Parsarthi, Wendy Mao, and this one. They are all here, and so I ask if Chris would like to say a few words about the.
Certainly. Where do I begin? So, I'll start with, again, this is number 2 this week. Very difficult time in the lab. 2 core members are leaving, and it's going to make a lot of work for me in the future. In Cesar's case, he's been one of the bedrocks for the lab as well as the building, helping out all sorts of side projects in addition to the actual project that you are seeing here. I'm going to try to keep this short because I could go on forever when it comes to Cesar. So you're welcome to stay afterwards to get all the highlight reels. But Cesar, I would say, is the best biomedical engineer all around that I've had as a student here, and no offense to Youssef, or to Simon if you're listening, and potentially future hire. But it's not your defense. And so Cesar took a while to get to this point. Many points along the way, I didn't know if he would ever actually get here. Cesar took some cajoling to sit down and start writing his defense because he loves to play around in the lab. If there's anybody doing any project, he will do that instead of sit down and write. So it took some cajoling to get him to sit behind a computer, start typing up all the wonderful research that you're going to hear about today. It also took a thesis defense delay, as well as an apartment lease to end, as well as a whole bunch of other things to get him to do it. And so I wish you luck.
So, thank you for the introduction, Dr. Shiva, Dr. Pasaglia. So today, we're going to get started with getting going. So, characterization of intracranial pressure and its relationship intracranial pressure and its relationship to intraocular pressure in the rat. So, what is this about? So the objective of this work is to characterize intracranial pressure variability and its relationship to intraocular pressure. And I'll define these terms in a little bit, so bear with me. But why are we actually even looking at this? So the reason that we're looking at this is because we want to gain more insight into what's happening at the optic nerve head environment and the pressure that could affect vision loss through optic nerve head deformations. So to get started, I want to define what intraocular pressure is, IOP. Intraocular pressure is the balance between aqueous humor production in the ciliary body of the eye and then out through into the anterior chamber and then exits primarily through the trabecular meshwork via Schlemm's canal and drainage vessels. So that is a primary pathway, the conventional outflow pathway. And then a secondary outflow pathway, which is the unconventional pathway, is through a lot of tissue diffusion here in the uveoscleral region. We're going to primarily focus on the trabecular meshwork pathway for this talk. And then the other critical thing that is necessary to understand for the rest of the presentation is what is intracranial pressure, or ICP. So ICP is defined as cerebrospinal fluid pressure, CSFP, in the inside of the skull, inside of the cranium. And so cerebrospinal fluid is produced in the choroid plexus here deep in the brain, and then it exits through various different channels, and then eventually encompasses the entire central nervous system. And this then goes out into the subarachnoid space, which is this little teeny tiny gap here between the brain and the rest of the meninges, then out through the arachnoid granulations and into the superior sagittal sinus primarily to drain out into the venous system. And so another key factor about CSFP, intracranial pressure, and intraocular pressure is that they meet at the optic nerve head. The optic nerve head is a structure in the back of the eye where retinal ganglion cell axons actually exit and become the optic nerve and allow us to transmit information from the eye and into the brain, giving us the ability to see. And this is where intraocular pressure pushes backwards onto this lamina fibrosa structure in humans, and then CSFP pushes forward on this lamina fibrosa structure as well. And so deformations to this lamina could be pathogenic and could cause the retinal ganglion cells to actually die and leads to ultimately vision loss. And the pressure balance is between the two compartments is called translaminar pressure, or TOP, which is conventionally defined as the intraocular pressure value subtracted by the intracranial pressure value. And in humans, normal intraocular pressure is between 9 and 19 millimeters of mercury, and intracranial pressure is between 7 and 17 millimeters of mercury. In the rat, which is going to be the animal model that I primarily do these studies on, it's going to be, IOP is typically between 9 and 17 millimeters of mercury, and 4 and 7 millimeters of mercury for intracranial pressure. And so what diseases are actually affected by these optic nerve head deformations? So in my lab, we primarily focus on glaucoma. Glaucoma is its primary risk factor is actually elevated intraocular pressure. So if you have high intraocular pressure, what tends to happen is you have this deformation of the lamina fibrosa, and that leads to this retinal ganglion cell death. And this cupping is what we see whenever you go into the ophthalmologist and actually get your eye checked. And that ultimately is one of the primary diagnosing tools we have for glaucoma. And then the antithesis to glaucoma is actually papilledema. So papilledema is a little less common, but this is the primary mode of action for papilledema is from elevated intracranial pressure. And this elevated intracranial pressure can come from various different disease states, such as traumatic brain injury, cerebral hemorrhaging, and potentially brain tumors as well, along with other cerebrospinal fluid disruptions. And so this is actually cupping of, or not cupping necessarily, but bulging of the optic nerve head into the eye, which also causes these disruptions in the axons of the ganglion cells, leading to vision impairments. And so now that we know the two primary, most prevalent disease states for this imbalance of pressure, what exactly are we going to look at? So I want to give you guys an outline of what we're going to be talking about today. So the first thing is going to primarily focus on characterization of ventricular and subarachnoid intracranial pressure in the rat, looking primarily at the variability, and then telemetric measurement of cerebrospinal fluid pressure and translaminar pressure in the pigmented rat. And then finally, we're going to be quantifying the effects of ICP elevation, so artificial ICP elevation on intraocular pressure and its related aqueous humor dynamics. And we're going to answer the following questions as we go along. So starting off with aim 1. What do we know about intracranial pressure and its relationship into the optic nerve head to begin with? This is kind of setting up our understanding of why I'm even looking at intracranial pressure in the first place. And so in a study done in dogs, you can actually see that ventricular intracranial pressure is highly correlated with the subarachnoid space pressure inside of the optic nerve, because the optic nerve, as I mentioned earlier, has cerebrospinal fluid surrounding it. And so this pressure is transduced from the ventricles out to potentially the subarachnoid space and then back into the eye. So this high correlation allows me to have a kind of an assumption of what's going on at the optic nerve head. And then the other thing that we need to know is that intracranial pressure is not a static waveform. Intracranial pressure is highly, highly dynamic, with oscillations ranging from seconds to slower oscillations ranging in minutes. And in a study done in mice with telemetry, we actually see that there's cardiac oscillations and within a 2-minute time scale shown by this asterisk here, and then respiratory oscillations, which is shown by this pound symbol in the waveform there. And then if we zoom out in the mice, you can actually see that there's these slower fluctuations that the authors of this paper attributed to animal activity. And then in humans, which is why we also are doing these studies, you can see that there's a lot of variability at various timescales as well. So the primary waves that are identified in the brain in humans are the following. They're Lundberg's A, B, and C waves. So the C waves are believed to be fairly normal. They would reflect respiratory and cardiac oscillations in the human brain. And the A waves are a medical emergency. So if this is usually found in patients with some sort of cerebrospinal fluid deficiency. And the B waves are actually a bit of a coin toss. It has been argued that these B waves are pathogenic, but in some other studies, people have seen that these are actually present under normal cases and cases of low consciousness. So the B wave is really what's the most interesting part here, because the first two are very well described, but the B wave is highly dynamic and it's has various different shapes, and we're gonna see what that looks like in the rat. Do these rats even have B waves? And so what we found is that the variability across the compartments that we're measuring here, so in the black trace, we have the ventricular space, which is deep in the brain here of the rat. And then in the red traces, we have the subarachnoid space, which is actually the envelope around the brain itself. And what we find at the really long time scales, this is 200 minutes, we see that there's these slow variations, but what I really care about at this time scale is the average, the mean. What does the mean value look like? And so what I found is that there's not a huge statistical difference between the mean, between the ventricular space and the subarachnoid space, though there is a difference where the ventricular space tends to be slightly higher than the subarachnoid space. And this slight difference physiologically could help drive cerebrospinal fluid out of the ventricles and into the subarachnoid space, although it's not statistically significant. And then another thing that you'll notice, I'm going to kind of jump around a little bit, is at the really large time scales, you see those slow oscillations, and then really fast time scales, you can see that there's these little wiggles, and these correspond to respiratory and cardiac oscillations, which is important to see in the brain and for proper measurement of cerebral health as well. But then in the middle here, these 4 plots that you see, you can actually see that there's a rhythmicity of these like spike-looking events. And they're too slow to be respiratory and too slow to be cardiac. And the rat, in these cases, is fully anesthetized. So what is actually causing it? So what is happening in this case? What do these oscillations even look like? And so zooming in a little bit more, we found that these oscillations are occurring at roughly the same exact rate as the human E waves, which is 1 to 2 cycles per minute. So in a 2-minute trace, we see 2 events. And because of this, moving forward, I'm going to call this the rat B wave. But the other thing that I wanted to know is, what is the shape? What is the characteristic shape of this event? And I'm recording from both compartments as well, and it's present in both compartments. So I want to first characterize the shape by taking the average of all of the events that were triggered by my detection algorithm. And what I found is that across various animals and over 700 events, we get one predominant waveform. And then this waveform was then modeled using this equation here. And you can see that the red line actually follows the black trace really well. The black line is the actual average B wave, and then the red trace is the model, the equation, fitted to the B wave in of itself. And we can see that it captures the overall shape really well, with some of the dynamics missing a little bit, but that could be further adjusted with more terms and more complex modeling, but we wanted to keep it fairly simple. And then the other thing that I noticed is that whenever you look at these events, there appears to be a time delay between the peaks. And this time delay between peaks is highlighted by these dashed lines here and here, where the black trace is the ventricular space B wave, and the red trace is the subarachnoid space B wave. And so I wanted to see how much of a delay there actually was across the various animals, and with that I did a cross-correlation analysis. And ultimately what that tells you is how similar these two waveforms are, and at what points do they actually match. And I found that there's a matched time point of about 1.2 seconds where the black trace actually leads the red trace, suggesting that the ventricular B wave happens first, and then it propagates out into the subarachnoid space, which is shown by this animation here, where the ventricles have the B wave occur first, goes out through the aqueduct, and then into the subarachnoid space where it's later detected. But this change is happening really fast in the order of seconds. And then the other characterizations of the waveform are that the ventricular B wave is larger in amplitude and shorter in duration. And then in the subarachnoid space, it is smaller in amplitude and longer in duration. And this is all approximated from the model parameters that we fit the waveform. But then this is normal. We're also interested in what happens if we take things out of the normal, out of norm. And so we did acute intracranial pressure elevations to kind of see what exactly would happen to these waveforms. Do they even change in shape? Do they change in occurrence? And what does that look like? And what I found is that the B wave itself and the variability does indeed change whenever you elevate intracranial pressure. And the B wave specifically, with the detection algorithm, actually become a lot more difficult to see visually. And so we need this mathematical model, this mathematical approach to not just visually try to figure it out. We want to have numbers tell us what is actually happening. And so in doing so, we were able to characterize the actual frequency of occurrence and the changes in amplitude between pre-elevation, peri-elevation, and post-elevation. And what we found is that while there was more events that occurred, more B waves that occurred during the ICP elevation case, the pre and post event counts were not significantly different from the elevation counts there. And then the other major finding is that the amplitude of the B wave itself does indeed change, where it increases in amplitude during ICP elevation, and this is true for both the subarachnoid space, which is in red, and the ventricular space, which is in black. And then finally, looking at the actual durations of how long these events last, we only saw a significant difference in the ventricular space, but the subarachnoid space did follow the same trend, though statistically it's not significant. But physiologically, these two track each other very well. And so, what are the key takeaways from this first aim? So to summarize, we know that ICP variability comes from fast and slow oscillations with the cardiac and respiratory. And then this is the first time that anybody has ever analyzed the B wave in a rat, which appears to be originating from the ventricular space out into the subarachnoid space. And then the other thing that we found is that elevating intracranial pressure actually causes mixed effects to the B wave, where it increases its amplitude, but it has a mixed effect in the duration, primarily affecting the ventricular space rather than the subarachnoid. And clinically, what does this mean? So this work suggests that the brown nori rat is a great animal model to study these B waves, which once again have been reported to be shown after traumatic brain injury in normal tension hydrocephalus, and it's also seen during a low consciousness states like sleep. And this is important because the B wave origin is unknown in humans, and its role in intracranial pressure health is also still highly debated. And then another key finding, looking at the multiple compartments, is that we found that the ventricular and subarachnoid space don't have much of a difference between each other in terms of the mean. So then, because they're very similar in the mean value, we can reasonably approximate what's happening at the optic nerve head. And that's because the ventricular space and the subarachnoid space are fluidically connected, and the subarachnoid space itself goes all the way down to the optic nerve. And that fluidic transduction should be relatively constant. And now that we have characterized intracranial pressure availability, let's look at it now in an awake animal. So in order to do so, we need to create telemetry devices. So my lab is really good at recording intraocular pressure in the rats. They have been doing it for many years with many devices, but intracranial pressure is a little bit of a new thing in the lab as well. And what prior studies have shown in just literature in general show that intracranial pressure is affected by various things. For example, in a rat study, you see that intracranial pressure is affected by the changes in posture, so whether the rat is nose up or nose down, and even is affected by jugular vein compressions. And then with the telemetry studies here, with the solid state transducer, you can see that there's a lot of different spikes happening, different events over many different timescales. And so the authors of these studies reference the animal activity to being some of the reasons for these spikes, though they don't have any video evidence that they had to correlate these events with. And then another major question that our lab is particularly interested in, because we look at diurnal changes and circadian changes in intraocular pressure, is does cerebrospinal fluid pressure itself have a diurnal rhythm? And the literature is conflicted. Some studies show that there is no diurnal rhythm. The ICP is fairly constant throughout the day and night cycles. And other studies, like the one shown here in this panel, have shown that there is indeed a diurnal change. So there's about a 1 millimeter of mercury change between light and dark in the study down here, and rats as well. But what does that mean for our rats? We use pigmented rats, these are albino rats, and we also don't particularly use the same age as they do. So who do we believe? And the question, to answer that question, I'm going to say neither, because I want to figure it out myself. And what are the implications about the potential actual the circadian rhythms or diurnal rhythm of intracranial pressure or lack thereof? And how does that affect translaminar pressure in and of itself? And like I said, I wanted to go and figure it out. And so I created a custom telemetry system to actually record intracranial pressure directly from the lateral ventricles. And as you remember from the previous aim, this is a valid approach because the subarachnoid space pressure is fairly consistent with what we see in the ventricular space. And I am the one that invented this device, but I'm also the surgeon. So I wanted to make my life easier whenever I do these surgeries, and I created a custom system to then just guide me and allow me to put holes wherever I need to do, wherever I need to put them on the skull, aligning them to these different structures on the rat's skull, and then ultimately combining everything in the assembly like you see in the first animation there. And then you can have an overall result with the rat wearing a hat throughout the experimentation phase. So what does this look like? Intracranial pressure or cerebrospinal fluid pressure, from my studies, does not appear to show any diurnal change. So it's fairly consistent throughout day and night cycles. And in order to test that, I did the mean, I looked at the differences in the mean between light and dark. I did find statistical significance, though doing further analysis, it's not a practical, physiological statistical difference, because it's only a change of about 0.8, 0.7 millimeters of mercury, which is relatively small between light and dark. And then another way to kind of test and really understand whether this change is actually relevant or not, I did what's called an autocorrelation. And really what you need to know from this plot here is that there's a lack of periodicity between 24 hours in the lifetime. So because there's no change, large peak here, I can then further conclude that there is no diurnal change. In the variability, there's also not a lot of change there from light to dark, though the dark phase variability is still higher, so there's more wiggles during the dark phase. And then looking at the distributions themselves of all of the animals and all of their different pressure values, you see that distributions overlap each other pretty well. So meaning that between the light and the dark, there's not a huge difference, and that physiological, those physiological values don't really mean too much in this regard. So from this I conclude that there's no diurnal rhythm in cerebrospinal fluid pressure. But how much of this variability, so going back to this variability, how much of this change in variability is due to various factors such as animal activity? Well, thankfully, I can figure that out myself too. I then changed the sample rate of my telemetry system, going from really slow samples, one sample every few seconds, to now multiple samples per second. And what I found is that when the rats are active, and this is all recorded with, these active and idle states are recorded concurrently with video, so that we can actually confirm that the rats are actually moving or they're just being idle. And so during the active phase, the rats have a higher intracranial pressure and compared to the idle phase. And then, as expected, the variability, the variance, how much it changes from the mean is higher whenever the rat is active compared to idle. And this is very obvious here in this top plot that you see. There's a lot more wiggles when the rat is active, and then there's less wiggles when it's idle. And then in this video, you can actually appreciate that a little bit more, where you see the rat was idle for a little bit. You see that the pressure is kind of stable, and then he like wakes up, moves his head around, and you see these large pressure spikes. But then again, the rat isn't always just on all its four legs. Sometimes it has to go up and grab some food, so it changes posture, goes up like this, or other times it's asleep and it has its head down. How much of that actually contributes to this variability? And that's a question that I wanted to answer myself as well, but I wanted to look at it in a controlled environment. And so I anesthetized these rats with isoflurane, and I created another apparatus to tilt the animals under this isoflurane anesthesia. And so by tilting them, I isolated any animal activity under, and looked at just the pure effect of tilting the rats. So at baseline, we have a relatively stable intracranial pressure waveform. And then the key thing that you need to take away from this is that when the rats are in the nose down configuration, here and here, here and here with a negative 90 and negative 45, you see these massive, massive changes in intracranial pressure. And then when the rats are nose up, you don't see the equal and opposite changes. And so one of the reasons that I think that you don't see the equal and opposite changes is due to venous return. So whenever us humans, we do a handstand, or we're like, hanging upside down on the monkey bars, we can feel that our faces are turning red. We can feel the pressure starting to build. And that is contributed to the amount of blood that's actually pooling on your head, and that is increasing this intracranial pressure in the rats. Whereas whenever the rats are up, and even us humans more upright, gravity is helping drive that fluid, that blood out of our head and allowing us to maintain a stable pressure as our heart is pumping blood into the brain as well to maintain that perfusion pressure. And then in doing these experiments, one thing that I noticed is that ICP was highly elevated under baseline conditions. So as you probably remember, the values that I was getting were between 3 and 5 and 6. This is 8 millimeters of mercury on average across different animals. And I had a hypothesis about isoflurane actually potentially elevating intracranial pressure, so I set out to test that as well. And what I found is that indeed, isoflurane does cause this mean change in intracranial pressure. And this elevation is sustained throughout the duration of the experimentation phase. And if you look at the actual time course of what's happening at those scales, this is the baseline trace where you can see that there's these wiggles, there's cardiac and respiratory oscillations, but then under isoflurane, you see this massive increase in the cardiac change. So you get every single time the heart beats under isoflurane, you get about a 4 millimeter of mercury peak to peak change. And then that is sustained at the 30-minute mark here, and then at the 60-minute mark here. And another anesthetic that I use a lot is ketamine, which is also the anesthetic that I use in the entirety of the first aim that I just presented. So I wanted to make sure that there were no confounds with any of my results there. And you can see that ketamine actually does not change intracranial pressure mean. It keeps it fairly stable, and which is good for my experiments in the previous aim. And then also, what does that variability look like? So this is the baseline once again. And then you can see that there is still this cardiac oscillation change, where the respiratory oscillations are a little pretty consistent between the baseline value and the ketamine value, but the cardiac oscillations don't increase as much as in isoflurane, but they are significantly higher in the 60-minute time point with ketamine and the 90-minute time point with ketamine. So the reason that these cardiac oscillations might be increasing is because both anesthetics have actually been shown to increase cerebral blood flow to the brain. And so with isoflurane, it appears it's a vasodilator, so it makes the vessels bigger. And that is why we see these, at least I think, why we see those cardiac oscillation changes. And then ketamine as well increases cerebral blood flow, and that causes these oscillations to get bigger as they have more of an impact in the intracranial pressure waveform. But now the other thing that I was curious about is my entire first aim was with ketamine anesthesia, and we found the B waves in the rats. And then the literature says that there is B waves under low consciousness states. So does this mean that every single anesthetic is going to give me B waves? And the answer is no. I ran the same algorithm, peak detection algorithm from the previous aim into these two cases, and for isoflurane, you see that there are no slow events that surpass the threshold. And the threshold is set by simply looking at how much variability there is, and then if there's anything that goes above that variability is what we call a B wave based on all of our work in the first aim. And then in the ketamine case, which I was hoping we would see B waves, and we do, thankfully, in 2 to 3 animals, is that you see that the noise level is lower during ketamine, but you can also see these slower peaks surpassing that threshold. And not every single peak is considered a B wave, which is denoted by the black dots. The B waves themselves that fit the model that we described in the previous aim are actually these green dots that we see there. And looking at the actual statistics of them, we see that the B waves actually look pretty similar in terms of amplitude and duration to what we determined in the previous aim with both the ventricular cannulation. Because I'm recording from the ventricles here, and I recorded in the ventricles in the subarachnoid space in aim 1. And so a potential cause for us being able to detect the B wave in ketamine and not isoflurane could be due to the mode of action of these anesthetics. So one anesthetic, isoflurane, stimulates GABA receptors, which is a primary inhibitory neurotransmitter. And so that is how it produces this anesthetic state. If you're sleepy, essentially, it makes you sleepier. And then for ketamine, it primarily inhibits the excitatory neurotransmitter. So in that example, in the sleepy example, if you're actually trying to fight sleep, you're being active, ketamine will tell you no, you're sleepy. So then it'll shut it down. And that's essentially what's happening, what I think is happening in the two differences with isoflurane and ketamine modes of action. And so we've answered 2 of the 3 questions so far for this aim. And now we want to transition into how this all affects translaminar pressure. Because remember, we care about the optic nerve. And so I had to develop another telemetry system. And this telemetry system is combining the intraocular pressure telemetry system we already have with some of the design elements of my new cerebrospinal fluid pressure telemetry system. And so in developing this, we're actually the first people to ever record translaminar pressure directly in an animal as small as a rat. And so the eye cannulation part to record intraocular pressure was done by my colleague Ali, and the brain part was done by me. And all of the analysis was then further done to look at this translaminar pressure difference. And what we found is that, to be expected, intraocular pressure, like I said, has this very pronounced diurnal rhythm. And it's low during the day and high during the night. Whereas intracranial pressure, as it relates to all of this, doesn't have this diurnal change either, which is consistent with what we found in the previous telemetry study. And what that means is that intraocular pressure is actually the main driver of this diurnal change in translaminar pressure. And as you can see, the statistics also follow suit, where light phase IOP is about 10 millimeters of mercury less than the night phase IOP, and that is very consistent with what translaminar pressure shows as well. And then, as you remember earlier, I had this autocorrelation where I had just a narrow peak and then long tails. Well, actually, in this case, we do see a very clear peak at 24 hours, both in positive and negative direction, and with a zero, with a high correlation at the zero plane as well. And so what does this mean for the optic nerve head in the rat? It means that these deformations could be happening throughout the course of a light and dark cycle. And how do these rats not develop any sort of glaucomatous damage, since IOP is the primary driving factor for glaucomatous damage? And a hypothesis that I have for this is that the glial lamina biomechanics is actually what's helping these rats not receive that level of damage to the optic nerve head, because the lamina fibrosa in humans and primates is very collagenous, so it's a lot stiffer. Whereas the glial lamina, as its name tells, is composed of neural tissue. It has glial cells there to help nourish and maintain the structure of the optic nerve head. And so because it's softer, I believe that it's able to withstand more pressure loads. And then, but that's not to say that they're not susceptible to glaucomatous damage. If you elevate intraocular pressure, sustained elevation of intraocular pressure does still cause this retinal ganglion cell death and the cupping and everything that you see in glaucoma in primates. So with that being said, I think that whenever you systemically elevate intraocular pressure, keep it high for a long time, you're not allowing the optic nerve head to return to its baseline value, and that lack of returning to its baseline is actually what's causing this glaucomatous damage as well. And my results are also in disagreement with the hypothesis that states that translaminar pressure should be consistent throughout the day to maintain this optic nerve head healthy. And so in the rats specifically, we see that that is not the case, where translaminar pressure fluctuates very drastically between night and day. And so to summarize this aim, what did we learn? So we learned that I made 2 telemetry systems to chronically measure cerebrospinal fluid pressure and translaminar pressure directly in pigmented rats. Cerebrospinal fluid is affected by animal activity, body orientation, and anesthetics. And the B wave occurrence is somehow correlated to ketamine anesthesia, ketamine and xylazine specifically, and that the translaminar pressure has a diurnal rhythm that is primarily modulated by intraocular pressure over a 12-hour light and 12-hour dark cycle. And now that we're on the topic of translaminar pressure, I also wanted to set out to see what exactly happens if you elevate intracranial pressure and how that affects the eye. So the literature states that if you stimulate the hypothalamus of the brain, so it's a very specialized structure within your brain, you get these changes in cardiovascular changes where your heart rate and your blood pressure are elevated. You also see, when recording intracranial pressure, that there's this elevation in intracranial pressure and then an elevation in intraocular pressure. But you'll note that they don't really track the cardiovascular changes very well, and also the intracranial pressure change, the maximum intracranial pressure change occurs before the intraocular pressure change. And so a potential explanation for that is actually from a study from our own lab where they mechanically elevated intracranial pressure, so whole brain elevation, and they found that aqueous humor facility, if you remember from the beginning, the trabecular meshwork resistance is the one that's changing. And so this could be a potential explanation for why this intraocular pressure change occurred, though the dynamics, the actual time course of that is still unknown, because these measurements take a little bit of a while to get, and you need multiple different intraocular pressure steps and flows to then regress that line, get the slope, which is your resistance, or facility, which is conductance. And then the other key finding with this is that applying tetrodotoxin, a neurotoxin that blocks innervation to the eye, you actually eliminate this effect. So the origin is believed to be neural. And because it's neural, there's some sort of control centers within the brain that could be affecting both ICP and IOP. And so what did I find? So when I elevated intracranial pressure, I get a bit of a mixed bag. However, on average, you see that intraocular pressure is actually changing in response to this intracranial pressure. And one thing to note here is that I have these different pressure steps, but I have varying durations of the ICP pulse. So in order to look at the overall shape, I normalize to the beginning of my ICP pulse, which is time 0, and the end of my ICP pulse, which is time 1. So it's all relative to the beginning and the end of the intracranial pressure pulse. And what you see is that there's a very characteristic IOP response, mostly in this green trace here, which has the highest samples. And this is a bit of a mixed bag because this was the least standardized stimulus parameter that I had at 15 millimeters mercury elevation above baseline. And then numerically what that looks like is I would characterize the time to peak of this IOP response across all of the animals that I did. And if we take the normalized time and actually put it in real time, I wanted to figure out how different the time course was from when I ended the stimulus to the actual time to peak of the IOP change. And what I found, and I call that the lag time. And what I found is that in the 5 millimeter mercury elevation case, I had a lag time of about 0.9 minutes or 0.89 minutes, meaning that the IOP pulse, or the IOP response peaked 0.9 minutes after I ended the ICP stimulus. In the green case, which is the 10 millimeter mercury elevation, it was about 4.2 minutes after the ICP elevation pulse ended. And then in the 15 millimeter mercury elevation case, it was about 5 minutes as well. And I'm reporting medians here, since the distributions are not normally distributed, and I'm also comparing through various different sample sizes. And so what we gather from this is that the IOP response is highly dependent on the duration of this intracranial pressure pulse, but the amplitudes don't really show much of a correlation between pulse amplitudes either. And now another set of experiments that I was curious about was looking at the amount of fluid needed to maintain intraocular pressure set at a 30 millimeters of mercury. And so the assumption was that if there were any changes happening, it would be detected by this flow, as if resistance was increasing, you would need less flow to maintain intraocular pressure at that level. And if resistance was decreasing, you would need to increase flow to maintain intraocular pressure at that level. And so what we found is on average that you need less fluid to be infused into the eye to maintain it elevated in all cases. Though you can see that there's a lot of variability, there's huge standard deviations, which are the shaded areas here. And what that looks like, or what that means, is that the facility could be what's changing here, and I'm actually going to be testing that in a little bit, but it's, like I said, it's a bit of a mixed bag. And when we look at the actual times to the maximum effect, or times to minimum, or times to maximum in some cases, you see that we get these negative lag times. And these negative lag times are a bit counterintuitive because it means that the eye somehow knows when the ICP pulse is going to end, which is not really the case. Physiologically, that doesn't make sense. But what I think is actually happening is that there is some sort of fixed time course that this effect reaches its maximum, and that the pulse duration goes beyond that maximum time that is needed. And so, for example, if the largest change is happening at 15 minutes pretty consistently, then a 30-minute pulse will give me a lag time of -15, whereas a 20-minute pulse will give me a lag time of -10. So that is where the normalization kind of breaks down a little bit, but we want to look really at the overall shape and look at the general response to this ICP elevation. And then another thing that I wanted to characterize was to see whether or not I can measure facility from this flow data. And I termed it a pseudo facility because it's not true facility, where I'm regressing multiple step points. I'm actually holding IOP at 30 millimeters of mercury and then measuring this flow term in the Goldman equation here. And then IOP is set, and so what I found is that my pseudo facility calculations actually are within the accepted value of what facility is in the rats that are age and species matched. And so another thing that I wanted to figure out was, can I then predict IOP at baseline? So this is before any ICP elevation happens. If I can predict IOP from this facility change, and indeed what I see here is that my cohort where I only measured IOP, is shown here, and then where I've calculated IOP from my facility values is shown here. There's no real difference between the two values there, and statistically they're not significantly different. And then as a final sort of sanity check to kind of make sure that I am actually calculating these values correctly, I then recorded intraocular pressure in one eye, and then I was holding pressure steady at that elevated level and measuring the flow, and then I calculated what the predicted intraocular pressure value, which is IOP star, it would be in the same exact animal, and there was no statistical difference between the two. So pseudo-facility measurements can accurately predict the real facility and IOP at baseline. And then the other thing that I wanted to test is how does this facility actually change in response to this IOP elevation, assuming that it's the only thing that's changing. And I expected to see something similar to this, but I wanted to see the actual time course. And what we see here is, using the average flow data, you see that there is this dip, and the values are actually pretty consistent with what is seen here, but the dynamics are very different in regards to the time course changes between each individual ICP elevations, and with the most representative being this green trace here. And then I wanted to see if I could predict what the IOP response for this flow change would be and try to match it to what the actual IOP response was, shown in the dashed lines. And in the 5mm mercury case, it tracks it fairly well, but in the 10mm mercury case and the 15mm mercury case, it doesn't track the dynamics well enough. It starts off pretty good in both cases, but then it kind of goes out of whack after a while. And so this tells us that facility might not actually be the only thing that's changing in response to ICP elevation. But I still wanted to know if this was a neural mechanism or not. And so I then applied this tetrodotoxin to the animals that showed an IOP response. And in both animals that I tested this on, I saw that the response was actually eliminated from this tetrodotoxin application. And in doing so, it confirmed that there is some neural feedback there, but facility from the previous results might not be the only factor that's actually being changed in response to intracranial pressure elevation, because they don't predict fully what the IOP response should have been. And so in conclusion, we find that ICP elevation elevates IOP to varying levels, and overall acute effects are dependent on the duration of the ICP insult. And then ICP elevation reduces the amount of flow needed to maintain IOP at a certain level, but it doesn't predict the dynamics of the IOP response purely using the flow rate changes, suggesting that there's other factors affecting IOP dynamics in response to ICP elevation. And then TTX application did eliminate this IOP response pretty robustly, and so it's still a neural mechanism that could be at play here. And this animation, if it would play, actually shows, well, it's not playing, but basically this animation, what it would show is intracranial pressure would elevate, it would send a signal to the eye, and then the eye would then respond to that signal, and that is the hypothesized neural mechanism between intracranial pressure elevation and IOP changes. So now we've answered all of these questions with all of the aims, and I want to leave you all with some final remarks, final overall messages. And so the key takeaway is that this dissertation is the first to characterize the rat B wave and propose the brown Norway rat as a model to explore the function and origins of the B wave. ICP or CSFP within the skull is highly dynamic. It responds to cardiac and respiratory oscillations along with animal activity and body orientation, along with different responses to anesthetics. And the optic nerve head environment is primarily influenced by IOP fluctuations rather than CSFP fluctuations. And then finally, acute elevations of intraocular or intracranial pressure do cause changes in aqueous humor dynamics and cause intraocular pressure changes, though facility may not be the only factor affected by this ICP elevation. And with that, thank you so much for coming today, and I want to thank my lab mates and my family and everyone here.
Before we open up to questions, questions, I wanted to sort of mention something I forgot to say earlier, and it would be a travesty to not bring this up. And that is that, you know, Cesar, when he gave this presentation, right, and the data just appears, and it all looks very straightforward, simple, and so forth. But the technical accomplishment here is why I said he's second to none, the best biomedical engineer I've had in the lab. Because, you know, normally what most people do to record intracranial pressure is they'll go in with a needle into this space, and they're measuring pressure at the same time. And they know that they've popped in to the ventricle because suddenly the pressure reading will jump, right? That's kind of more or less what he did in his first experiment. But this thing, this is a whole new beast. It's like, it blows me away, really, the technical accomplishment here. Because he's got one shot to do this. So to do this right, he's got to, you know, he made that little stencil he told you about. So he's got to put that in the right location, so it has to be all mapped out perfectly, because you want to hit the fattest part, because it's not even thickness. You want to hit the fattest part to get that needle in that location. So he's got one shot, it's all blind, right? Because he basically makes a little hole, sticks his needle down, and everything has to be made to perfection, so about 5 millimeters, right? What's it, 5 millimeters, is that much depth, right? That needle has to end exactly in that spot. And that spot is about the thickness of your toenail, right? So, and it's amazing to me. Like, it works almost every time. So it is really a technical accomplishment.
With that, thank you.
Thank you.
The floor is open for questions.
Alright, so to get Chris that excited about science like this, I think this would be really something. I don't know, thank you, this was great talk. So I have like two kind of shameless questions back to
Sure.
that. The first one, is there any potential that you have thought about that you could use like ICP or cerebrospinal fluid pressure as like targets for treatment in glaucoma or other disease? Or are there non-invasive ways that you can modulate these?
So you're asking about whether intracranial pressure can be used as potential therapy for glaucoma to stabilize that cupping?
Yeah.
So that could be a potential, we could explore that potentially, but there have been studies that have actually looked at already doing that, and it's more in the relationship of space-associated neuroocular syndrome. And in those studies, they found that whenever you elevate intracranial pressure to then prevent any of this glaucomatous damage, there's not really any significance there. There's not really too much happening in terms of neuroprotection, because at the end of the day too, if you have glaucoma pushing back on the eye this way, and you have intracranial pressure pushing on the eye this way, you're kind of squeezing that structure, and so you might be actually causing more damage to that structure rather than actually helping it by maintaining that cupping to a minimum, because you're still squeezing those structures at that space.
And also kind of connected to that in research aspect. So you showed that by elevating intracranial pressure, you could get somehow like an elevation IOP.
Mm-hmm.
Do you think that elevation in IOP is sustainable, and whether that could be potentially used as like a glaucoma model in animals?
So the IOP elevation in and of itself may not be fully sustainable, because these are acute experiments, and so what I think is happening is that there's some level of feedback that is happening between the eye and the brain, and if you keep ICP elevated, I think that there's going to be some sort of changes that IOP is going to want to come back to normal. And the level of ICP elevation, or sorry, not the ICP elevation, but the level at which intraocular pressure changes in the studies that I've done so far is about 3 millimeters of mercury. So that is not a super large change in IOP to potentially cause this glaucomatous damage. And though if it's sustained, it might eventually cause it, but you may need to sustain it for a very long time, whereas most like microbead injection models or even the hypotonic saline models cause like 10 millimeters of mercury, 20 millimeters of mercury changes sustained, which cause that glaucomatous damage.
Do you think like using another method to crank up that IOP?
What was that?
You think you can't use something else to like, really increase the IOP elevation after messing with your ICP?
That's a good question. I don't think there's really anything else to really crank it up aside from just going in and sticking a needle into the eye and just elevating it with like hypotonic saline or artificial aqueous humor.
Yeah.
Question?
You got a question from Dr. Derek Lura. Was there any filtering or frequency analysis of the raw ICP signal?
Absolutely. So thank you for that question, Dr. Lura. So when we're looking at the ICP signal, I'm a nerd, so I appreciate a lot of these really complex mathematical things, and I wanted to present this in such a way for everyone to understand. But yes, when looking at these cardiac and respiratory oscillations, we did frequency analysis, so power spectrum analysis, to actually show, and I think I have some supplemental slides here, to show that these events are actually happening at these specific frequencies, which correlate to a specific rhythm, a specific rate. So for example, a peak here in this 1 hertz band is actually corresponds to respiratory rate, which is about 1 breath per minute, or 1 hertz. And then this one here is at 4 hertz, this peak, which corresponds to heart rate, which is about 240 beats per minute in a rat. And so, yeah, we did some filtering, we looked at the actual waveforms themselves and characterized them further. And then to isolate the B wave, we actually low pass filter to get rid of all of these extra things and then isolate those slow oscillations to then allow us to perfectly view what is actually happening without all this extra wiggles in the waveform. Any other questions?
If you were doing one more year for your PhD, what extra would you do?
One more year in my PhD, what actually would I do? I would actually work more on the instrumentation of the translaminar pressure, because that's the main idea within all of this, and put all of that from the back onto the rat's head, and then record intracranial pressure and intraocular pressure directly there, to then be able to then, at high frequencies as well, to then correlate all of these different effects that I see in ICP with the IOP waveform as well, and then really understand and get that insight into translaminar pressure and what is actually happening in the rats.
Very nice talk. I wanted to ask, I saw you had 2 different ways of measuring ICP, one with the head mount right on the head and one that was on the back. Do you notice, is there any differences between the 2 systems, or are they?
Very good question. So there were some differences between the 2 systems, primarily because one system, the one that's on the head, is fully rigid. So most of the variations that we see from that system should be endogenous to what's happening in the cerebrospinal fluid compartment, with some inertial effects as well. Whereas the backpack system has this tubing that's fairly compliant, so it's able to bend and able to cause these potential motion artifacts that could affect the variability. And in terms of the mean, the backpack system was lower than the cerebrospinal fluid system, though they were not super distant from each other. Yes?
Very nice talk.
Thank you.
And amazing technique.
Thank
I
you.
was always amazed about what you are doing.
Thank you, thank you.
Yeah. So that whether there are publications or whether you can predict whether there's the ICP pattern change when the IOP increase, so like in control patients. So yeah, the ICP pattern change when the IOP is elevated.
So do you think, so the question is whether IOP elevation affects ICP backwards?
Yeah.
So the answer to that is no. We found that in our lab and throughout various studies where when you elevate intracranial intraocular pressure artificially, there is no signal going to the brain telling it to elevate as well. It's only a one-directional signal that can be eliminated with the application of this tetrodotoxin. And so the rationale for that, I believe, is because there's actual structures in other studies that, I wanted to put a lot more here, but I only have limited time. But in other studies, they show that there's actual neural projections going from the anterior structures of the eye down into the sympathetic nervous system and then all the way up to the hypothalamus with retrograde labeling. So that mechanism is potentially originating in the hypothalamus, going out through the sympathetic nervous system and into the anterior chamber of the eye. So, but there's no evidence to suggest that IOP elevation actually causes an ICP change. That answer the question? Sweet. Any other questions?
If there are no other questions, once again, thank you. I request the committee members to stay here and the others, please leave.